Hello and good afternoon everyone. My name is Ella Mead and I'm an Associate Analytical Research and Development Scientist at Owlstone Medical. And today I'm going to be introducing to you Owlstone's Breath Biopsy VOC Atlas. I'm going to begin by talking you through what is the Breath Atlas and then how we have created the Breath Atlas. So, what is Owlstone's Breath VOC Atlas? It is an ever-growing list of VOCs that exist on breath, differentiated from background contamination and found repeatedly in a human population. Next, the question is, what are the benefits of Breath VOC and Atlas? Firstly, knowing what a VOC on breath really is provides an understanding of the underpinning biological pathways which enables us to really understand how breath VOCs behave in different populations. This aids to better study designs and speeding up of biomarker validation. And additionally, knowing exactly what is on breath helps us to target the performance of those compounds during optimization of key processes in the sampling platform, such as breath collection, sample storage, analysis, and feature extraction. So, how did we create the Breath Atlas? At Owlstone, we've used our in-house Omni platform, which is an end-to-end process that consists of three main steps, which is collection, analysis, and interpretation. The breath and blank samples were collected using our receiver mouthpiece sampler with a Casper air supply, and the thermal desorption used for sampling onto contain a multi-sorbent bed consisting of a weaker sorbent, 10-ax, and a stronger sorbent, carbograph. Then, for analysis, the samples were stored, dry purged, and then analysed on the Mark's TD Thermoscientific Q-Exactive-Orbitrap system. So our analytical instrumentation has been optimised to maximise the potential for detection of VOCs. Our thermal desorption sample introduction equipment includes a cold trap, which contains the same sorbent material as our sample tubes, and the GC column has been chosen to optimise the retention and peak qualities of these VOCs we detect on each breath sample. And we have used a TD-GC-Orbitrap SIL-MS column. And our high-resolution Orbitrap mass specs allow for thorough deconvolution of the data using Thermo's Compound Discoverer software. So through analysis of our equipment and room blanks in conjunction with the breath samples, we employ a methodology to eliminate any peaks that may arise from various sources. Our primary metric we use for on-breath detection is our standard deviation method, where we compute the mean average signal for a specific feature within the equipment blank. So, to be considered an on-breath VOC feature, the signal intensity must surpass the threshold of the mean plus 3 standard deviations of the equipment blank signal in at least 50% of the breath population. So this filters out any potential noise or background fluctuations in the data. Our secondary metric we use is a paired t-test approach, which considers only the corresponding equipment blank sample of the breath sample and calculates whether the difference between the two is significantly different by performing a paired t-test with a p-value of 0.05 and a fold change of at least 2. And finally, our third metric of on-breath determination is using our receiver operator curve area under the curve. Applying all three of these on-breath metrics, a total of 585 features were considered to be on-breath from our dataset containing nearly 1,500 features. So out of these 585 features initially identified to be present in breath using our three metrics, we were successfully acquired 568 VOCs for our compound ID workflow. And from this subset, we were able to incorporate 432 VOCs into our high-resolution accurate mass library, ensuring compatibility with our analytical method. And subsequently, our process has confirmed the presence of 154 of these compounds to be in our breath samples. So, to conclude, the Breath Biopsy Atlas is a compendium of on-breath VOCs observed in a diverse cohort of UK adults. Atlas will serve as a reference dataset to facilitate biomarker discovery projects and currently consists of 154 compounds with multiple biological pathways being represented. Future work will expand the breadth of the biological pathways and chemical classes included in the Atlas. I'd just like to take a moment to say thank you to the team who have worked really hard on this project and continuing to do so. And just quickly mention our Breath Biopsy conference, which is coming up at the start of November. You can register for free at owlstonemedical.com/bbcon. Thank you very much for listening. If you have any questions, I'll be at poster 44.